Welcome to Stanford University's inaugural 3-minute thesis competition. I'm Ken Goodson. My name is Ken Goodson, and I'm the Stanford Vice Provost for Graduate Education. It's fabulous to see such an amazing crowd for this event. Thanks for being here. I want to say a quick word of thanks to our on-site volunteers who've helped you get to your seats on time. So as you see on the screen, the 3-minute thesis competition was launched at the University of Queensland back in 2008. And since that time, it has been adopted by nearly 1,000 universities worldwide. We're delighted to give it a special Stanford spin today, and it's going to be very exciting. It challenges PhD students to distill their scholarship into 3 minutes for a broad audience. That's us today. It's an incredibly important skill to reach people across the country with what you're doing. It's a great thing for us to be working on and for these students to be working on. And it's a privilege and an honor to bring us all together in this moment with everything that's happening in this country to celebrate graduate research. How about a hand for that? Okay, we've got some rules that the finalists who've been preparing really hard, the rules that the finalists have to follow. Each finalist has 3 minutes, and it's really strict. Those who exceed 3 minutes will be disqualified. The timer starts at the first word spoken. And when it comes to time, I want to introduce our timekeeper, Ken Shu. Ken, please stand. Ken is the Assistant Vice Provost and Director of the Graduate Life Office. Now, Ken has a lot of jobs. Not only does he start the timer, but with 30 seconds left, he has a placard. And that's right, you'll see it. And with 10 seconds left, he has a placard. And then there's a red placard, and you know what that means. Okay. Back to the rules for the finalists. Presentations must be spoken. No poems, no raps or songs. It's spoken narrative. And each finalist has designed a single static image that you'll see on the screen, a visual element to complement what they're telling you about. No animations, no additional electronic media. There are no props or costumes. It's just the student and 3 minutes. So there is one actually fun detail. Each finalist has selected a musical clip which will accompany them as they come on stage to build the excitement. And that'll be fun for all of us. Okay, judging. We are really delighted to have a panel of very distinguished judges today, and I'm going to introduce them. If you could please stand when I call your names. Condoleezza Rice. She is the Tad and Diane Taub Director of the Hoover Institution and a professor in multiple departments here at Stanford. David Studdert. David is the Vice Provost and Dean of Research here at Stanford and a professor of health policy in the medical school and of law. Michelle Rasmussen. Michelle is our Vice Provost for Student Affairs. Martin Schell. Martin is Vice President and Chief External Relations Officer here at Stanford. W.E. Murner. He is the Mosher Professor of Chemistry and Professor, by courtesy, of Applied Physics and the 2014 Nobel Laureate in Chemistry. So the judges will be evaluating our presenters on content, of course, but also engagement and communication with the audience, and that's all of you. There will be awards for first, second, and third place at $5,000, $3,000, $1,000, and there's a People's Choice Award, and that's all of you, and you'll get instructions, but be taking notes because you will get to weigh in on the winner of the People's Choice Award. And we have some rules in the audience as well. You can cheer, yell, and make as much noise as you want between the 3-minute segments. And so we hope to have a very festive time between the segments. But when the finalists are speaking, we ask for absolute pin drop silence. They have a very strict timeline. They're under a lot of stress. Please give them your full focused attention. And please silence your phones. So, we are very happy to have a master of ceremonies, top-notch this afternoon, our 13th president, President John Levin, who will be taking the stage to introduce the finalists.
Okay, good afternoon. Thank you for being here. It is a joy to host this inaugural event. As it goes on, I'm going to share a bit about how this event was developed, including how the finalists were selected and prepared. However, we have 10 of them to get through. So without further ado, let's go straight to the first finalist. First up, we have Jody Lunger, PhD candidate in genetics, and her presentation is called Just Eat It, Engineering Our Immune System to Fight Cancer.
Like many of you, I know someone who has faced the frightening reality of cancer. Like many of you, I know someone who has faced the frightening reality of cancer. This highlights why advancing cancer treatment is still one of the main goals of modern medicine. Now, we've come a long way in the last century, but we still need new approaches to treating cancer. An ideal approach includes 3 things. First, it needs to efficiently find the tumor. Second, it needs to accurately detect cancer cells. And lastly, it needs to eliminate the cancer without harming healthy tissue. It turns out we actually already have a system in our body that's fully capable of doing just this. I'm talking about the immune system. Cells of the immune system patrol our body, protecting us from outside threats such as bacteria and viruses, but also from internal threats such as the rise of cancerous cells. The problem is, in a cancer patient, tumors have learned to hide from the immune system, rendering immune cells inactive soldiers when they should be at war. My thesis aims to re-engage this army, allowing immune cells to once again fight and destroy cancer. The star of the show is an immune cell we call the macrophage. Macrophages are special in part because they can leave the bloodstream and penetrate deep into tissues where tumors can hide. Secondly, we can now engineer these macrophages to contain proteins on their surface that specifically recognize cancerous cells. Lastly, once that macrophage finds its cancer cell target, it then goes on to simply just eat it. This unique mechanism of macrophages allows them to eliminate cancer without destroying healthy tissue. But of course, there are some challenges. Tumors can be big, and macrophages are only so hungry. In our lab, we've developed a second protein. This protein acts like a command center inside the cell, turning on signals and telling the macrophage to perform certain functions. Through my research, I've shown that by equipping macrophages with this second protein, we can dramatically increase their ability to eat cancer cells at a much faster pace. We also hope to use this protein to push the macrophage to send signals to other immune cells, recruiting them to this tumor site, turning a lone fight into a coordinated attack. By engineering macrophages to make a tumor its next favorite meal and attracting other immune cells to the cause, I'm excited to be contributing to progress in immune cell therapy, what I believe is the next frontier of cancer treatment. Thank you.
All right, thank you, Jody. That was terrific. Okay, so while our judges are considering the presentation they just saw, I want to take just a minute to tell you a little bit about the finalists. This is obviously an amazing celebration of Stanford graduate students and their scholarship, and this has also been a tremendous professional journey for them. To just become a finalist, they had to be chosen from dozens of submissions from all across the university. The competition was fierce, and what you're going to see as we go through this is that all the different schools, many different departments, and many different disciplines are represented among this group. It's a very wide breadth of scholarship. And with that, I'm going to bring up our second finalist, who is Favor Nuriz. She is a PhD candidate in electrical engineering, and her presentation is titled Quick Reflexes and Lost Memories, Teaching AI to Spot Brain Disease. Favor, come on up.
Here's to the ones that we got. Cheers to the wish you were here, but you're not. Cause the drinks bring back all the memories of everything we've been through. Toast to the ones here today. Toast to the ones that we lost on the way. Cause the drinks bring back all the memories.
Have you ever wondered how fast your reaction time is? Let's find out. On the count of 3, clap once as quickly as you can. Nope. Not yet. On my count, 1, 2, 3. That quick reaction, a perfect harmony between your brain and body, is something we often take for granted. But however, there are millions of people with Alzheimer's and Parkinson's disease whose slowdown becomes a daily reality when they can't do such reactions. Simple things like walking smoothly, clapping your hands, or remembering these wonderful faces become increasingly difficult. Right now, doctors rely on very expensive brain scans and invasive tests that only provide a brief snapshot of what's happening in your brain and body. It's like trying to understand an entire movie's plot from just one pause scene. Clearly, we need a better way. My research addresses this gap using 2 powerful tools: artificial intelligence, AI, and everyday physiological signals. I measure physiological signals like brain activity using a portable EEG headset, your heart rate activity using your smartwatch, and your walking pattern using video recordings. Then I train AI to act like a skilled detective that constantly spots clues in changes in someone's daily movements, their speaking patterns, or even their typing speed. It learns what normal looks like for each person so it can detect tiny shifts long before they become noticeable symptoms. But I'm not stopping at looking at these signals individually. I'm also exploring how these signals interact with each other to provide a richer picture of the brain's communication network. Imagine your brain and body as musicians in an orchestra. Usually, they're perfectly synchronized. In Alzheimer's or Parkinson's, though, some instruments gradually fall out of sync or stop playing altogether. By tracking these disruptions, we can spot early signs of disease progression a lot sooner. This early detection matters enormously because it means interventions like drugs and other treatments can start sooner to reduce the damages diseases can cause. Imagine being able to track your brain health as easily as you can see your daily steps or even knowing your heart rate activity right from the comfort of your home. Okay, now let's finish with one more synchronized count. On my count. Ready? 1, 2, 3. Next time you clap, whether cheering at a game or celebrating a loved one's celebration, remember, these are the moments I'm working so hard to protect. Thank you.
Okay, thank you, Favor. Another great presentation. So the finalists, just to tell you a little about how this whole process worked, the finalists in this event have not only been working on their dissertations, their thesis research for a number of years. They've been in this process for several months. They had to submit videos back in January, and then they've been practicing and rehearsing and memorizing and working with coaches in the oral communications program and participating in various run-throughs. And they probably are running through their scripts in their sleep, actually. But hopefully they've learned a lot from this process as well. I'm now going to bring up our third finalist, who is Lydia Burleson, who is a PhD candidate in English, and her presentation is titled Two Sides of American Utopia.
In the same boat with a lot of your friends, waiting for the day your ship will come in.
I'd like you to guess when this quote I'm about to share was written. America is not merely a land or an institution. America is the nameless foreigner, the homeless refugee, the hungry boy begging for a job. All of us, native-born or alien, educated or illiterate, we are America. She is still our unfinished dream. Carlos Bulosan wrote these words in his 1946 novel, America is in the Heart, voicing a utopic vision that was so strong that he immigrated from the Philippines to Seattle in 1930. A vision that is so enduring that these words feel like they could have been spoken in our own political moment. She is still our unfinished dream. Is this not what the American Liberal Party speaks for when they support the rights of the country's most vulnerable? Is this not what the American Conservative Party speaks to when they talk about keeping this country great? I research utopic visions of the early 20th century and their resonances in the present. I argue that we can use the novels, images, and texts of the previous generation to better understand our current divisive moment, a moment where, much like the 1920s, two very different visions imagined a better world for the US. On the one hand, we have a vision like Bulosan's, texts that speak to this investment in American utopia through the fight for workers' rights. This is a pro-immigrant, multiracial, working-class vision, a vision where all Americans are given equal rights and protections as American workers. On the other hand, there was another vision, a vision that was so compelling that Stanford's first president, David Starr Jordan, was one of its strongest supporters. The eugenic vision, now debunked, made use of pseudoscientific discourse and advances in evolutionary theory to push for a certain kind of ideal American, and that everyone else was biologically inferior. You can see this discourse mobilized in Bulosan when, upon reaching Seattle for the very first time, he was immediately described by a white traveler as a monkey. Another race, another class, another type of person. My research looks at how these two competing visions collide. And though they may seem ideologically opposed, I argue that these visions are actually intimately connected, and that we can see those connections in descriptions of marginalized Americans, from the Black life that Richard Wright cataloged, to the Filipino Americans in Bulosan's text, to the poor rural white families who are also subject to eugenic analysis. By looking at these shifting discourses on race and class in a period of immense economic change, I argue we can better understand the interconnection of these discourses in our present, in the process gaining a new lens to work on that unfinished American dream, a dream that will always be a work in progress. Thank you.
Okay, thank you, Lydia. I think one thing you're going to see in this set of presentations is the just incredible range of disciplines and work that takes place across the university. And our next finalist is going to expand that even further. Our next finalist is Allie Singleton, who's a PhD candidate in the Emmett Interdisciplinary Program in Environment and Resources, and her presentation is titled Highways and Hidden Epidemics, the Unseen Cost of Development. Come on up, Allie.
We've built over 400,000 kilometers of highway worldwide, a length equal to driving around the world 10 times. But how do we decide where to build highways? Highways are critical for connecting us to jobs, family, and new opportunities. They also have unintended consequences, such as noise, pollution, community division, and, in tropical forests, deforestation. But there's one impact both policymakers and researchers have thus far overlooked: highways' impact on infectious disease. 2 summers ago, I traveled along a highway in the Peruvian Amazon. The highway was built in 2009, and since then, residents there have reported a troubling trend: rising cases of dengue virus. Dengue is transmitted by the bite of an infected mosquito and is the fastest spreading mosquito-borne disease worldwide. There is no vaccine. But how would a highway impact dengue if it's spread by mosquitoes? Although mosquitoes aren't very good flyers, humans can travel far distances, especially with the help of a new highway. I hypothesized that the newly built highway allowed people infected with dengue to travel more quickly throughout the region, seeding outbreaks along the highway corridor. To quantify the impact that the highway had on dengue transmission, I collaborated with local health departments, analyzing dengue case data before and after the highway was paved, while controlling for other potential changes, like population growth. The results were striking. The highway caused an additional 10,000 dengue cases in a region of only 100,000 people. While dengue is rarely fatal, it can have devastating economic impacts. As important as this highway was, could we have done better? Now, I'm scaling this research to assess the impact of the Amazon's entire highway network. With multiple large-scale highway construction projects planned in the region over the next decade, my findings suggest that these projects could up to double the amount of dengue transmission in the entire Amazon basin. But this isn't just about dengue. The approach I've developed can also be applied to other diseases like malaria, Zika, and even HIV. Highways are critically important parts of our world's infrastructure, but they're also spreading disease. My research reveals a previously hidden cost of this infrastructure in tropical forests and offers tools to help policymakers weigh and mitigate these consequences. By considering health impacts alongside economic benefits, we can make smarter, more sustainable decisions for our planet and its people. Thank you.
Okay, thank you, Allison. By the way, the music that all of the candidates selected, that was the final stage of the selection process. They were first selected through this initial process, but then at the last moment they had to pick what would be their walk-on music. By the way, doesn't everyone want to have some walk-on music when you just sort of like go like you're coming up to bat in a major league game or coming out to pitch in relief or something? So let me say now our 5th finalist, Gabe Amador. Gabe is a PhD candidate in developmental biology. His presentation is titled, "Must Be This Tall to Ride. Plant Stem Cells Act According to Their Size." Okay, come on up, Gabe.
When you look out over the Stanford campus, most of what your eye sees is a dense patchwork of plants, living, breathing, ever-growing plants. Plants like these provide the food on our plates, the wood on these walls and ceilings, even the very oxygen we breathe. I'm a plant biologist, and to me, one of the most amazing facts about plants is that their bodies are built mostly out of air. The heft of a tree comes not from the soil, not from the rain, but from carbon dioxide that's pulled in from the atmosphere. To absorb all that carbon, plants use these tiny pores that cover every surface of their leaves. And you can see them. If you look at a growing leaf under a microscope, you'll see a picture like that one on the top left with 2 kinds of cells, a sea of immature stem cells in gray, and among them in purple, those tiny pores through which carbon dioxide enters the plant body and oxygen escapes. It's through those pores, numbering in the billions or trillions in a typical tree, that plants breathe. So, how are these pores made? The object of my dissertation was to understand how that sea of immature stem cells gives rise to these pores. We started this project by simply looking, and we noticed something funny. It turns out that among all of those stem cells, the only ones that ever turn into pores were the smallest ones. That observation led to a hypothesis that the cells may be on a sort of cycle, illustrated here, where on each pass, a cell starts by checking its size. And if it's a big cell, it keeps riding that cycle, divides, gets smaller, tries again. But if it's really, really small, it exits the cycle and turns into a pore. So that was the idea, but in science, ideas are the easy part. What's hard is testing them. To test this model, we turned to a favorite tool in the biologist's toolkit, the genetic mutant. For a biologist, a mutant isn't defective, it's an opportunity to change one aspect of biology and observe the consequences. In our case, we found mutants that change the size of those stem cells. And just as our model predicted, if you make them really big, they stay on that cycle for much longer. But if you start them out really small, they come in, check their size, and exit on their very first pass. So by changing the size of these cells, we can control their behavior and thereby the production of these pores. Our work highlights how out all around us on this campus, and in forests and farmers' fields, and in our own gardens, tiny little stem cells are making decisions about how to build a robust and functional leaf. And I believe that in a future where food security and climate resilience depend on smarter and more adaptable crops, we can use these discoveries to not just understand the behavior of stem cells, but to control how plants breathe and grow. Thank you.
Okay, thank you, Gabe. Now, there were many exceptional entries to this competition, and so it was quite hard to narrow it down to 10. We want to acknowledge a number of entries that were exceptional, but just didn't make it to the final 10. And so there are 12 other theses that were selected for honorable mention, and they are up here on the slide. And if you're one of those 12, please stand up so we can applaud you. Okay, and with that, let me bring up our sixth finalist, who is Catherine Bowman. And Catherine is a PhD candidate in epidemiology and population health, and her presentation is titled Lymphedema, Cancer Care Complication, or the Sinister Side of Survivorship.
When I was a little girl, I made a promise to my mother that would set off a chain reaction in my life path. After all, she, like 30% of other cancer survivors, had just developed lymphedema as a result of her cancer treatment. Now, lymphedema is a lifelong and incurable disease of the lymphatic system. This is a key part of our immune system and our ability to manage our swelling. We can think of the lymphatic system a bit like a straw that's filled with water. When a cancer patient goes in for treatment, say a surgery to remove lymph nodes with cancer in them, it's almost like cutting that straw in half. One part of the lymphatic system begins to leak out fluid, while the other part struggles to pick it back up on the other side. This results in lifelong swelling of the arm and a decreased ability to transport our immune cells to fight infection or other threats. Now, as I saw my mother's quality of life change with her lymphedema, I made a promise. I said, "Mom, I'm going to cure you one day." And as a teenager, I began to work on this promise, but I noticed every year or so, my mom would develop a serious skin infection and even a skin cancer at the site of her lymphedema. Little did we know, these were early signs of my mother's changing immune system. Now, I tell you this story because it really sets the stage for my thesis work. It begs the question, what if lymphedema has more sinister effects beyond changing quality of life alone? Well, by mapping how often a breast cancer survivor with lymphedema is actually experiencing something, say, like a cancer reoccurrence, or seeing how long that they actually live past their diagnosis, and comparing this back to a counter-reality where these patients never developed lymphedema at all, we can calculate whether lymphedema meaningfully increases the risk of cancer reoccurrence or even death amongst breast cancer survivors. Within my own thesis work, I've started to tackle these calculations related to the immune system. For example, I've demonstrated that breast cancer-related lymphedema patients are almost 3 times as likely to develop those skin infections my mom encountered when compared to breast cancer survivors without this disease. This result now prompts me to ask 3 more important questions in my thesis about the immune system of these patients.
whether lymphedema patients are dying earlier, if they're dying earlier due to cancer recurrence, and if so, what factors clinicians might look for in determining their patient's risk of that recurrence. Practically speaking, my thesis work helps us to understand a potential opportunity to improve the lives of breast cancer survivors through early identification of lymphedema. It also helps to ensure that my promise is kept not only to my mother, but to all cancer patients encountering a lymphedema diagnosis after their treatment. And finally, it helps to give a voice to the 250 million patients worldwide living with lymphedema by exploring a new potential impact of this disease on the health of our population. Thank you.
Terrific. Thank you, Catherine. I just would ask you to notice, in addition to the breadth of disciplines that are represented, the amazing, just in these presentations alone, the incredible span of research from essentially sort of pure curiosity-driven additions to knowledge all the way out to patient care. And that is really a hallmark of the university, that the research is both broad and spans so much of the pathway from just advancing knowledge all the way out to changing people's lives. There's one group I want to thank before we go to our next finalist. And I mentioned before that once the students were selected to be in this competition, they had a chance to work with communications coaches. And they got to work together in small groups to work on their scripts, to work on their presentations, to just get feedback. And this is our team of communications coaches. And if you're one of the people in this group, I'd ask you to stand up so that we could acknowledge you and give you a round of applause. Thank you for the great work to get these presentations ready. Our seventh finalist is Michaela Reby, and a PhD candidate in chemistry and pathology, and her presentation is titled Shining the Spotlight on Cancer's Sweet Tricks.
Imagine you're a detective trying to catch a bad guy, but this culprit is wearing the perfect disguise, blending in so well that even the best security system can't spot it. Cancer does the same thing, staying undetected by our body's best defense, the immune system. Normally, the immune system is designed to recognize and destroy threats like cancer. However, cancer uses camouflage tactics, manipulating its surface to blend in, staying undetected by our body's best defense. Every cell in our body is covered in sugar, kind of like the chocolate shell of a peanut M&M. This sugar coat acts as a secret code that helps cells communicate with each other. Cancer cells take advantage of this by altering their sugar coating to blend in. This defense is able to stay undetected and unrecognizable. My job as a detective is to crack the code of this sugary disguise. And to do so, my research uses a new approach, creating specialized probes based off of the immune cell detectors themselves so that we can determine exactly what it is the immune system is seeing. These probes are able to decipher the sugary cloaks of cancer cells, revealing the hidden messages that they send in order to avoid immune attacks. By using this technology of immune cell-based detector probes, I work with clinicians from around the world to analyze samples of human tumors with the latest imaging techniques. And what my findings show is that it's not all sugars, but specific patterns of sugars that play a key role in helping cancer hide and spread. And these patterns appear in countless types of different cancers, such as cancers of the colon, prostate, breast, and brain. In these cancers, I've detected up to 5 times amount of these sugar patterns as compared to healthy cells. By decoding these deceptive patterns and connecting them with the way cancer progresses, we are able to expose and target these disguises. This not only uncovers cancer, but opens a door to innovative ways we can empower the immune system to mount a strong and effective defense against a cunning adversary. Cancer is a master of disguise and may sugarcoat its crimes, but with this research, we will shine a spotlight on its sweet tricks.
Thank you, Michaela. Okay, as a reminder, when the presentations are over, you are going to get to vote on your favorite presentation for the People's Choice Award. So make sure you're keeping your own notes. And then when it ends, we're going to collect your votes through a QR code. So the voting will open at the end of the event, and you'll hopefully have your phone, and you'll have 5 minutes to vote. So just keep that in mind that that's coming up. And now I'm going to get to our 8th finalist, who is Tamri Matiashvili, who's a PhD candidate in economics. And her presentation is titled Talent, Trust, and Health: How Women Changed Medicine. Come on up, Tamri.
In 1872, medicine was revolutionized. The Russian Empire opened the first formal medical school for women. Letting women become doctors transformed medicine and saved lives. But how, and at what scale? In my research, I studied some of the first female physicians who lived over 150 years ago. To do this, I had to face 2 fundamental challenges. First was finding the data. I digitized documents from libraries in 3 different countries, covering over 350 counties of the Russian Empire over 35 years. These records track the numbers of male and female physicians, healthcare infrastructure, and vital statistics like births and deaths, all in rural counties of the Russian Empire where each doctor mattered. Importantly, I was able to pinpoint the year in which a county hired its first female physician. The second challenge was identifying the causal effect of hiring women on health outcomes. I used the statistical method called the staggered difference in differences design, which compares changes in health outcomes in counties that hired a female physician in a given year to changes in those outcomes in counties that had not yet hired a female physician in that year. I find that female physicians reduced infant mortality by over 140 deaths in an average year in county. But why were female physicians so much more effective than the male physicians? First, letting women become doctors allowed some of the most talented women to redirect their potential to healthcare. Health outcomes improved for everyone. Young men and women died less, and so did hospital patients. Smallpox vaccines increased. Second, women started going to doctors more. In patriarchal societies, women would often avoid seeking care from male physicians out of shame or mistrust. When counties hired their first female physicians, their reliance on untrained midwives decreased, while births in hospitals increased, making it safer for both mothers and children. Overall, I find that female physicians improved health outcomes for everyone. But unfortunately today, due to inequalities in both education and the workforce, there are still many countries with very few female physicians, especially in rural areas. And as my research shows, alleviating these inequalities is imperative for all of our health and well-being. Thank you.
Some of you might be wondering what motivated our finalists to compete. So we did ask the finalists, why did they decide to do this? I mean, everyone who is a PhD student here is working incredibly hard on their research. That's basically all you do as a PhD student. You just work all the time. I'm actually not joking. That really is all you do as a PhD student. So we asked, why did you want to participate in this event? And actually, there were a lot of similarities in their response. And what they basically said was they really love the work they're doing, and they're very proud of it. And it's exciting to get to share it with a large audience like this. And it's also, I think, something that many people learn over time is to be able to communicate your work. When you, particularly in PhD, you're just narrowing down to the most specialized knowledge, and to be able to broaden out and figure out how can you communicate that out to a broad audience in a way that excites them about research is an incredible skill. So it's really, having this event here at Stanford is really a nice way to give this group of students a chance to go through that experience of working on communicating out the work that they do. Okay, with that, I want to bring up our 9th finalist, who is Sarah Jobalia, who's a PhD candidate in computer science, and her presentation is titled Hair Flow.
Is that India? What happened to her hair? What happened to her hair? What happened to her hair? Little girl with the press and curl.
I get my curls from my mother, and the thickness of my hair from my grandmother on my dad's side. My hair represents who and where I come from, my culture, and how I want to be perceived. What stories does your hair tell about you? Do you see hair like yours in animated movies or video games? Your answer will depend on your hair type. My name is Sarah Jobalia, and I study computer graphics, which is the math and physics behind computer animated movies and video games. Think Pixar style, rather than old school hand-drawn like The Lion King. My specialty is computer-generated hair. I can tell you that this difference in representation across hair types is not a coincidence. It's due to a technological gap. Have you noticed that almost every computer-animated black character before 2020 is either bald or wears a hat? When computer scientists first made hair, the field was heavily white, so they made straight hair. Soon, studios began to see curly and afro-textured hair as difficult and expensive, just because it hadn't yet been researched. Only after 17 years did computer scientists build a tool that would make curly hair at all for movies, for Brave. Now good artists can make afro-textured hair, but only with difficulty. My research builds a system that allows users to input a photo of themselves and get out a full 3D hairstyle they can use for their own projects, regardless of hair type. So how does my work give us something that looks like this to represent our input photo, where the best previous work can do is this uneven bob on the left? Well, let's first talk about what this hair is made of. 3D characters, like Merida here, have hair that is comprised of a bunch of points in 3D space. That's X, Y, and Z space. If you put a line through these points and connect them, you get a strand. Now do this 100,000 more times, and you get one head of 3D hair. My project uses machine learning to predict where these points should go in 3D space. This is easy enough for straight hair because the hair all moves together. The computer can predict this direction and then use it to place the points. But curls change direction a lot, way too much for the computer to understand, which is where previous work gets it wrong. Instead, we look at the center line of a curl, or the flow of the hairstyle. Then, using expertise from Black-focused hairstylists, seasoned computer animators, and hair biologists, we recreate the curl around our center line. Now our curls are back and accurate. With hairstyles like these, artists can make the characters kids like me wish I had growing up. Now, even new artists have the power to tell anyone's story.
Thank you, Sarah. Hopefully there are a few doctoral students in the audience right now who are thinking about doing this next year. I want to encourage anyone who is thinking about it to do this. We have one more finalist, and that's Kristen Abels, who is a PhD candidate in chemical engineering, and her presentation would be the final presentation. is titled From Brines to Batteries, Membranes for Critical Mineral Recovery. Come on up, Kristen.
Do you by chance own a smartphone? If so, congratulations, you own at least one lithium-ion battery. In truth, lithium batteries power a lot more than just our phones. They're powering the clean energy transition. They provide energy storage for renewable wind and solar power, and they're critical to the rapid expansion of the electric vehicle market. Nowadays, we're even calling lithium white gold because our clean energy future depends on it. And just like with gold rushes of centuries past, people are flocking to the lithium mining sector in anticipation of a skyrocket in lithium demand as we shift towards a low-carbon future. Here's the catch, though. Mining lithium from traditional rock and water sources is typically very chemical and energy intensive. Even with battery recycling, recovering 1 ton of lithium product can generate up to 10 tons of chemical waste. So this begs the question, if we're sourcing lithium with the goal of supporting the clean energy transition, shouldn't our mining and recycling practices align with this goal as well? I'm working to make lithium recovery just as sustainable as the rest of the clean energy transition by developing greener membrane technologies. Whether we're trying to extract lithium from a salty water brine or recover it from a battery waste stream, the main challenge remains the same, and that is separating lithium from a very complicated mixture to produce a very high purity product. To put this into perspective, imagine yourself in a giant ball pit, where 1 in every 10,000 balls is red. And your job is to filter through everything and collect only the red balls. This is analogous to the task of extracting dissolved lithium ions, the red balls, from a mixture of many different dissolved salts in a brine or battery waste stream. Not only does a very low lithium concentration make this a monumental task, but the very similar properties of the lithium ion to other ions in the solution make it very difficult to figure out ways to separate out just the lithium. While current techniques rely on either adding lots of chemical inputs or lots of energy inputs to drive this separation, I'm researching membranes as a greener alternative. My work includes designing new membrane materials with just the right Goldilocks chemistry to allow lithium ions to filter through while blocking the passage of other ions like a strainer would. And it also includes designing creative ways that we can use these membranes to displace current energy-intensive technologies. For example, I've worked with colleagues to design a membrane process that can reduce the energy intensity of a lithium extraction step by as much as 20 times. So next time you pick up your smartphone and question all the work that goes into sourcing the lithium that it uses, rest assured that researchers like myself are working hard to ensure the lithium supply chain is just as sustainable as the clean energy future that it permits. Thank you.
Terrific. All right, thank you, Kristen. I'd actually like to invite all of the finalists to come up on stage so we can give you one more big round of applause. So come on up, finalists. Okay, congratulations to all the finalists. That was amazing. Those presentations were so great, and the work that went into them was just magnificent. Can't wait to, see you defend your theses and go out and publish the papers. So that was that was really, really amazing. And let's give one more round of applause to our amazing presenters.